Hi, I'm Jaden, and today I will be talking about organ development and programmed cell death. Programmed cell death, or apoptosis, is a process that is crucial for development, tissue homeostasis, and immune function. The purpose of it is to eliminate unwanted cells. The dysregulation of apoptosis can cause cancer, autoimmune disorders, and neurodegenerative diseases. In 2002, Sidney Brenner, Robert Horvitz, and John Sulston won a Nobel Peace Prize in physiology or medicine for their work with organ development and programmed cell death. These are the credits for this video. Most of my information regarding the three men I found on the Nobel Peace Prize archive website. Along with this are the other websites I got the pictures and diagrams from, including MRC Laboratory of Molecular Biology, Understanding Animal Research, the Cleveland Clinic, Oncobytes, and NCBI Bookshelf. Sidney Brenner was born on January 13, 1927, in Germiston, South Africa. He died on April 5, 2019. At the time he won the Nobel, he was affiliated with the Molecular Science Institute in Berkeley, California. His work made it possible to link genetic analysis to cell division and organ formation. Brenner was the one who decided to use the nematode as the model system for this work because they are multicellular yet relatively simple. They are also transparent, which helped Brenner follow cell division directly under the microscope. Brenner used ethyl methane sulfonate to induce mutations. EMS induces mutations by alkylating guanine bases, primarily causing C to G pairing to transition to an A to T pairing, as seen in this picture on the right. The mutations that he found when conducting these experiments could be linked to organ development. Robert Horvitz was born on May 8, 1947 in Chicago, Illinois. He is still alive today at 77 years old. At the time he won the Nobel Prize, he was affiliated with the Massachusetts Institute of Technology, or MIT, in Cambridge, Massachusetts. In 1986, he identified two of the genes needed for programmed cell death to occur. He also identified genes that regulate how dead cells are removed from the body. Horvitz was the one who identified and mapped the mutant phenotypes that Brenner found using EMS. He then was able to map the mutant phenotypes to specific genes in the nematode genome. Horvitz discovered that the genes SED3 and SED4 are essential for programmed cell death. He also discovered that a specific gene, SED9, antagonizes this process in the nematode. The diagram shows the process of cell death using SED3, 4, and 9. As you can see, they are all active during the killing part of this diagram. John Sulston was born on March 27, 1942 in Fulmer, United Kingdom. He died on March 6, 2018 in Stapleford, Cambridgeshire, United Kingdom. His affiliation at the time he was awarded the Nobel was the Wellcome Trust Sanger Institute in Cambridge, United Kingdom. He discovered that certain cells' deaths were a part of the organism's normal development, as well as a first mutation in a gene that is active in the cell death process. Sulston drew out the dividing cells and then mapped out the sequences of cell changes and divisions in the nervous system. By drawing the cells, he revealed that specific cells undergo programmed cell death during development. He was also able to identify the mutations that disrupted this process. And this picture is a picture from his journal where he drew the cells and their mutations. The Nobel Prize website posted this picture because it was used by the three men to illustrate cell lineage in the top left and programmed cell death in the bottom of the picture in nematodes. It is described as, quote, "The fertilized egg cell undergoes a series of cell divisions leading to cell differentiation and cell specialization, eventually producing the adult organism in the top right of the picture." In C. elegans, All cell divisions and differentiations are invariant, i.e., identical from individual to individual, which made it possible to construct a cell lineage for all cell divisions. During development, 1,090 cells are generated, but precisely 131 of these cells are eliminated by programmed cell death. This results in an adult nematode composed of 959 somatic cells. Unquote. Understanding cell death during development can have a large societal impact. Since dysregulation of cell death can lead to diseases occurring, understanding it could help doctors prevent dysregulation from occurring altogether. Understanding PCD pathways can also lead to the development of new therapies that target PCD to treat diseases such as cancer, autoimmune diseases, and neurodegenerative diseases. This first picture shows how apoptosis is being used to target cancer cells. Specialized proteins are used to start the breakdown of important cell components and blebbing of the cell membrane. The other picture shows the stages of apoptosis and the benefits of it, including replacing old or damaged cells, strengthening immune systems, and supporting balance. Here are all of my credits in MLA9 format. Thank you for watching. I hope you enjoyed my presentation and learned something new.